that develop an alcohol use disorder throughout their life. And a recent study from the Lancet showed that this was in the beginning of the lockdown in the UK, that there was a sudden increase of 30% or over 30% in alcohol sales during COVID-19. So it may be that people, due to increased anxiety and stress, or maybe even boredom or not having a job to go to anymore, definitely increase their drinking. So it's very timely to look at alcohol use disorder. And there's an urgent need for interventions that can promote long-term abstinence as well as reduce alcohol craving. So my general research objectives are first to study the neurobiology of alcohol use disorder using neuroimaging to explore targets for treatment. And also I have and will utilize neuroimaging to capture effects of treatment in alcohol use disorder. And just in one slide to summarize what I have done so far, or what my interests are. So mainly during my PhD and postdoc, I've worked on the dopaminergic system, mainly functional MRI and alcohol cue reactivity and striatal reactivity to alcohol cues. And I've tested the effects on a behavioral intervention on this paradigm and showed lower amygdala reactivity after retraining of attention of alcohol on alcohol cues. Plus, I've worked on the dopaminergic system in terms of PET with carbon-11 reticuloprid. I've worked on or showed lower D2 receptor availability in smokers compared to non-smokers, and in cocaine users compared to non-users, and have also shown that chronic sleep deprivation may actually mediate this decrease in D2 receptor availability. Then I've worked on neurogenetics. Mainly, one of my interests for a while was the methylation as a biomarker of PET binding, mainly in the dopamine transporter. And also I've worked on genetic ancestry, as well as socioeconomic status and how this was associated with dopamine D2 receptors, actually. Then the first 2 years of my postdoc at NIH, I worked with PBR28 to work on neuroinflammation in alcohol use disorder, with a very strong hypothesis that those with an alcohol use disorder who were daily drinkers for years and years would have increased markers of inflammation, thus increased levels of TSPO, therefore increased PBR28 binding. However, we found the opposite. We actually found lower PBR28 binding, and there was another group at Yale. Ansel Hilmer was the lead author of this paper, who found exactly the same. So we followed up on that to see if this actually reflected lower inflammation in people with alcohol use disorder in a rat study. But in the rat study, we actually found or no difference in inflammation or using this PET marker, but post-mortem we actually did find elevated PBR28. So that was a very puzzling finding, and it has been published already 2 years ago, but definitely there we saw a mismatch between PET imaging in vivo versus in vitro. And last, something that I'm mainly working on now is brain metabolism in alcohol use disorder, both with FDG as well as with other labeled components or ketone bodies, and with magnetic resonance spectroscopy. So in this talk, I will first present the findings of a project that I've worked on in the last 3 years at NIH. That was part of my K99 study under the mentorship of Nora Volkov, namely on brain metabolism and alcohol withdrawal. Then I'll present my proposed study for the R00 on brain metabolism and alcohol consumption. And then recently we submitted a bench to bedside proposal. So it's a small research grant between an extramural and an intramural lab. And we propose this on brain metabolism and alcohol tolerance in collaboration with the NIDA lab of George Koop and Leandro Vendoscolo. And last, I'll discuss future directions at Penn. So first, the study that I've worked on in the last 3 years, entitled The Effects of a Ketogenic Diet on Alcohol Withdrawal in Alcohol Use Disorder, which was part of my K99. And finally, last Saturday, I submitted this for publication, which is nice. Now I can finally move on with the new studies. But yeah, several observations from the Volkov lab as well as other labs have shown that alcohol decreases brain glucose metabolism acutely, whereas it increases brain acetate uptake. So lower brain glucose metabolism has been shown already in alcohol use disorder, and was always thought that those are just have lower cognitive function because of this. But this study that came out in 2013 showed, well, rather than glucose, it may be that if the brain is under the influence of alcohol, acetate, which is a metabolite of alcohol, actually increase in the brain and may be used as an alternative energy fuel than glucose. And also interestingly, it was shown that heavy drinkers compared to social drinkers also had an elevated acetate metabolism in response to alcohol. So their brains may have been used more to this transition of acetate metabolism. And well, also during sobriety, so not only when one is intoxicated, but also during sobriety, it has been shown on the left with MRS that there's a higher acetate uptake in heavy drinkers compared to social drinkers, whereas on the right, many studies have shown lower brain glucose metabolism in those who regularly drink compared to those who don't. And this may be relevant to alcohol use disorder treatment, in that when somebody stops drinking, or we hypothesize that there may be a flux from high acetate.
I'm watching a video, Chris.
Okay. Oh.
Yeah.
Okay. Well, there may be a flux.
Yeah, people may want to mute their audio unless they're asking a question.
Oh yeah, please do ask me questions though, if you have one.
Yeah.
So there may be a flux or a transition when people quit drinking, namely from high acetate to low acetate, and from low glucose to high glucose, just from glucose in one's diet. And maybe this transition may lead to neurotoxicity and enhanced neuronal excitability in alcohol use disorder, and may contribute to the withdrawal symptoms that are present in those who quit drinking after years. And we thought that a ketogenic diet, which is a diet that is very high in fat but low in carbohydrates, and that has been often used for reducing seizures in epilepsy, that this may actually help people during withdrawal, because this diet leads to a state of metabolic ketosis, namely cells in the body are using ketone bodies rather than glucose, and these ketone bodies can either be acetoacetate, beta-hydroxybutyrate, as well as acetone. So not only our cells in our body, but also our brain can use these ketone bodies as an energy fuel. And there was an animal study that came out in 2018, but apparently also one that in 1981, when we actually really looked for it, that showed that this diet, a ketogenic diet, high-fat diet, may actually lower the symptoms of alcohol withdrawal in animal studies. So in the 2018 study led by Anders Fink Jensen from Denmark, he showed that those rats who were fed a ketogenic diet, it lowered body rigidity as well as irritability during detox compared to those who were on a regular diet during alcohol detox, and compared to those who were never drinkers. So the aims and hypotheses for our human trial were first to assess the effects of a ketogenic diet on withdrawal symptoms measured with the CIWA, which is just a behavioral questionnaire. as well as benzodiazepine use and alcohol craving. And we hypothesized that a ketogenic diet intervention would reduce alcohol or withdrawal symptoms, as well as the need for benzodiazepines, as well as craving. And second, we tested the effects of the ketogenic diet on ketone bodies in the brain using MRS. And there we hypothesized that ketones in the brain would mediate the effects of the diet on withdrawal and craving. So for the methods, we did this in the alcohol inpatient unit at the NIAAA in Bethesda in Maryland, where people are regularly being treated for alcohol use disorder, and their treatment program is about 3 to 4 weeks. So within 2 days of people arriving, we randomized them into either a ketone diet or a standard American diet. The standard American diet is lower in fat, but higher in carbohydrates and protein for 3 weeks. And I found it very important that this would be double blind, but how do you do that with a dietary intervention? That was a little tricky. I'll actually explain that in the next slide. And we looked at daily withdrawal symptoms in the first week of acute withdrawal, as well as craving ratings over 3 weeks. And every week we did an MRI scan where we measured or MRS with a press sequence, very standard sequence, and functional MRI alcohol cure activity, which NROs like so much as well. And for the MRS, we primarily looked in the dorsal anterior cingulate cortex, so a gray matter voxel. But yeah, so we randomized people into the KD versus standard American diet, and these diets were isocaloric, so they both had the same numbers of calories. And to make this blind, the metabolic unit of NIH developed a largely liquid diet that was either ketogenic or control, and it was very, very difficult to tell the difference by using, the control diet definitely contained sugar, and the ketogenic diet mainly had sweeteners. But we did a taste test among ourselves in the beginning of this trial, which started 3 years ago. And among our staff, we could not tell the difference between the two diets. So for breakfast, lunch, and dinner, patients received the shake mainly. And then on the side, they all received snacks that were ketogenic diets. So there's this lovely looking egg here on the right and all the other menu options. And patients definitely had trouble sticking to this diet, but they were monetarily rewarded for sticking to it for the full 3 weeks. So the final sample actually, the trial was still ongoing last March, but had to be stopped due to COVID-19, and we just decided that even though we wanted to have 25 versus 25 people in the study, we now have 19 versus 14, but mainly because of my move to Penn, we just wrote it up as is. So 53 people were randomized, 20 people were excluded or withdrawn due to various reasons. Some people really could not stand the diet. There was no difference, by the way, whether it was the keto diet or the control diet, but also some people just dropped out already on the first day because they had other interests. They didn't even want to be in the alcohol inpatient unit anymore. The final sample, 19 on the ketone diet and 14 in the standard American diet. And the two groups had relatively equal sample characteristics, and particularly in terms of drinking. There was a small difference though in BMI, namely there was one extreme outlier actually in the standard American diet that was very large, but there was no difference in weight loss after 3 weeks. So yeah, because they were receiving the same number of calories. So if we look at the results, every morning an independent nurse collected a urine sample of the participants, and in urine you can measure acetoacetate, so you can really easily see if somebody is in ketosis or not. And already 3 days after detoxification, which was roughly 2 days after the beginning of the trial, those who were randomized into the ketogenic diet group were already in metabolic ketosis, whereas those who received sugar in their shake, standard American diet, SA, did not, this did not lead to metabolic ketosis. And you see a small dip in the third week, and that was because 2 patients actually cheated. They could not send it anymore. They cheated for 2 days because they had a pizza party. But we still included them in it. We warned them, and they stayed on the trial. And very interesting, last week I spoke with Mackenzie Cervenka, who is a collaborator who works primarily on the ketogenic diet in patients with epilepsy, and she's a co-author on this paper, and she said, wow, I've never seen people going into ketosis this fast. So unfortunately we didn't have a control group of healthy volunteers, but it is definitely something I would like to follow up to see if those with alcohol use disorders, maybe because they're already used to acetate metabolism, maybe their body goes into ketosis faster than non-dependent healthy volunteers. And on the right, we see beta-hydroxybutyrate serum levels. Namely, every week there was a blood draw, and also there, those who received a ketogenic diet showed evidence that they were in ketosis. Then in the brain, we also showed acetone as well as acetoacetate concentrations in those patients who were in ketosis in the dorsal anterior cingulate cortex. We actually also wanted to measure beta-hydroxybutyrate, but we could not do that with this very standard press sequence. So this is also something I'd like to follow up. I've just discussed this with some collaborators at Yale to see if we can have a very an edited sequence that actually focuses on BHB rather on all metabolites. But still, we saw evidence of ketosis also in the brain. Then after the 3 weeks, we asked patients, so which diet did you think you were on? Did you think you were on the ketogenic diet or on the control diet? And there was no difference between the diets. So there we can conclude that the randomization was successful and the blinding was successful. But interestingly, there was a very small effect on an interaction of diet times diet expectation for pleasantness, namely those participants who thought they were on the ketogenic diet rated it as more pleasant than those who thought they were on the standard diet. But overall, those who completed the trial were pretty okay about it, but many people dropped out before. Then we looked at benzodiazepine intake. So in the left graph, we see oxazepam, which was mainly prescribed for alcohol withdrawal symptoms. And after the diet started, or there was a diet or group times time interaction effect. So those who were on a ketogenic diet needed fewer benzodiazepines. And the psychiatrists, or there were several psychiatrists prescribing this, they were all blind to the study intervention. And also on the right, if we look at only the cumulative dose, there was a trend level difference in lower oxazepam use in milligrams in the keto group compared to the standard American group. And also when we looked at the number of people who needed benzodiazepine intake at admission, when people were not randomized yet, there was no group difference between those who needed benzos or not benzos, whereas on day 2, when people were, or actually day 3 after admission, there was a difference. So this was when people were already in ketosis. Namely, there were fewer people on the KD who needed benzodiazepines. Then we looked at alcohol withdrawal, and there you see a very small effect, but it did not lead to a statistical difference. And it may be that's so those subjects where the CIWA or the withdrawal scores were larger than 8. So when there were actual withdrawal symptoms, only they received benzodiazepine. So the lack of effect here in withdrawal symptoms may actually be because those on the KD received fewer or less treatment than those on the standard American. So this may have led to this lack of an effect. Then we looked at the desire for alcohol questionnaire. every week, and we saw a trend effect only on this, namely somewhat lower alcohol craving in the keto group compared to the control group. And every week we did this alcohol cue-induced brain reactivity paradigm where inside the 3 Tesla MRI scanner, people saw food cues, alcohol cues, and neutral cues for 33 or 750 milliseconds. And after that, people also rated the cues for both wanting and for wanting alcohol cues as well as the food cues. And it was an interaction effect for the wanting of alcohol cues. It's a negative scale though. It's an alcohol cues minus neutral cues, which shows that participants explicitly rated that they did not want the alcohol cues, but still the keto group wanted alcohol even less compared to the standard American diet group. And this effect was not the case for food cues. And then there was also a group time interaction in the dorsal anterior cingulate cortex. We have more hypothesized that the ventral striatum would respond less to alcohol cues after the ketogenic diet intervention, but we actually showed elevated DACC activation to alcohol cues compared to neutral cues, as well as to food cues compared to neutral cues. And this region is implicated in self-control and the processing of salience attribution. So it may be involved, but it was definitely not an a priori region that we hypothesized. We also, with this MRS press sequence, we also have other metabolites that we looked at. And one of them is glutamate, very often seen to be elevated in those with alcohol use disorder compared to controls. And those on the ketogenic diet group actually increase glutamate as well as glutamine compared to those on the standard American. And glutamates can be used also as an energy fuel, and this is how we interpret it for now, but it was also not necessarily something we predicted, but still an interesting finding. I've never seen any study on the ketogenic diet on glutamate, so it was hard to also discuss this finding. And then we looked at myo-inositol and choline, which are measures of neuroinflammation, and there we saw a main effect of ketogenic diet, namely lower neuroinflammatory markers in the keto group compared to the standard American group. And one actually confounding thing is that the week 1 MRI scan, ideally we would have done a scan on the day of when people came in to the unit to have an actual baseline. But we couldn't do it. We first had to screen people, and also many people were still nauseated and not ready to do MRIs. So those who had the week 1 MRI done were already in ketosis. So it's quite difficult to actually look at a real baseline in this study. So a summary of this study, this was the first, well, to my knowledge, the first KD intervention in human alcohol use disorder, and its very, very rigid 4-to-1 ratio of fat to carbohydrates was relatively well tolerated. The diet lowered benzodiazepine intake, and therefore there was no effect on withdrawal. Also, the ketogenic diet reduced wanting of alcohol cues and alcohol craving at trend level. And the diet increased acetone and acetoacetate in the anterior cingulate cortex and elevated reactivity to alcohol cues, indicated enhanced control over alcohol. So many thanks for this study to the Volkov lab as well as the George Koob lab, which I'll explain later. We had an active collaboration on an animal study that went in parallel. Second, the question remains whether metabolic ketosis also has an impact on alcohol consumption, and that is part of my R00 project. So does ketosis influence alcohol consumption? We wrote this up actually, and rodent study is part of this first study that we just submitted. And in this study, which was led by Leandro Vendruscolo, he is a staff scientist in George Koob's lab at NIDA. So he worked with 36 Wistar rats, and for a week these Wistar rats learned how to self-administer alcohol, which apparently they can learn quite easily. They quite like it after a while. And then for 2 months, these rats were split up into 2 groups, namely 18 were being fed a ketogenic diet and 18 a chow diet. And here you can see below in the graphs that those on the keto diet, they had reduced blood glucose levels and elevated blood ketone or the beta-hydroxybutyrate levels, similar to the human trial and also similar as what you would expect. And then after those 2 months, all rats were being exposed to a chamber for 14 hours a day and either exposed to alcohol or to air. So half of the groups, half of the ketone groups and half of the control groups were being made alcohol dependent just by passively vaping alcohol for 7 weeks. And here you can see in panel B that during the alcohol vapor, there was actually no difference in blood alcohol levels for those who received the alcohol vaping in the rats who were previously fed a ketogenic diet or a chow diet. So there was no biological difference in the way they broke down alcohol. However, when they looked at alcohol self-administration, they saw that, and this is in dark blue, that those animals who were fed a chow diet or a regular diet, they escalated their drinking when being also presented to alcohol vaping. So those were the rats who were fed a chow diet, they were being made alcohol dependent, and they self-administered alcohol the most compared to the rats that received a ketogenic diet and were also made alcohol dependent. So there we concluded that a history of ketogenic diet lowers alcohol consumption. And well, the next step is also to look at those who are actually on the ketogenic diet. And Leandro did a preliminary trial on that, but he saw that those rats who were actually on the diet could not tolerate alcohol at all. So he's still working on that. But it's definitely an interesting finding as well, that while you're in ketosis, that apparently you cannot tolerate alcohol very well. But I don't have any graphs on that because he didn't record this. He just saw that they were all laying down very sadly and could not tolerate the alcohol. To be followed up, definitely. So for the R00 proposal, I aimed first to test whether metabolic ketosis decreases alcohol consumption in or AUD outpatients or just individuals with AUD, so human individuals with AUD who are not treatment seeking is probably a better way of saying it. And here I would hypothesize that ketosis would lower alcohol consumption. And second, to measure the effects of metabolic ketosis on brain ketone bodies again. And again there, I would hypothesize that the ketones in the brain would mediate the effects of ketosis on alcohol consumption. And rather than looking at this difficult to maintain ketogenic diet, my eye fell on this paper that came out in 2018, which is when I wrote the R00, or a project where a nutritional ketone ester, and this you can actually buy just over the counter at this company called HVMN, and it's an ester that you can drink. It's a very bitter solution, but not too bad. I don't mind it too much. But this increases metabolic ketosis in humans to a level actually similar to a 2-week ketogenic diet. And this study was done in Oxford. This particular ester was developed by Kieran Clark and also by Richard Veach, he passed away earlier this year, but who was an investigator at NIAAA as well. So I contacted him, and he advised me also on what to do in human AUD. But this study looked in particular to in healthy volunteers at the desire to eat. So it showed that The ester actually lowers the desire to eat just regular food. And also it decreased ghrelin, which has been associated with hunger levels, as well as actually with alcohol craving. So, and also here on the left, we see again the figure that I showed earlier, namely the beta-hydroxybutyrate levels in blood. And on the right is the pilot. It's actually me drinking one of those bottles and taking my own blood. And there you see that after half an hour, I actually reached a similar beta-hydroxybutyrate level in my blood compared to all those poor patients on this very rigid diet for 2 weeks. So, those levels are comparable, and I think for a study in individuals with AUD that is outpatient, it is definitely an advantage to work with this solution compared to the diet. So the proposed ROO design is to test 20 AUD patients that are non-treatment seeking and 20 healthy volunteers, and they would come to the lab, well, actually 3 times, first for a screening visit, of course, and then once where they receive a ketone ester and once where they receive a placebo, and every individual would receive both substances. And the study visits would, first they would receive this ketone ester or the placebo. Then immediately after, I would like to do an MRI scan of 1 hour. And after that, an alcohol self-administration paradigm to see whether it decreases alcohol self-administration. And during the MRI scan, I would like to do just a standard T1 and T2. Again, in functional MRI, alcohol cure activity, and again, MRS. But this time, rather than doing this very standard sequence, I would like to work together with the Yale group on this edited sequence that can actually look at BHB. So there I would hypothesize that in those patients with alcohol use disorder, that this ester would actually lead to a faster elevation of BHB in those patients compared to controls, similar to the PET finding previously. And after that, they would do an alcohol self-administration paradigm. I initially proposed a very basic implicit taste test. So, for example, just have a tray with alcohol and say, well, the substance that you've taken previously will change your taste, and please let me know what you like about this. And then what we actually testing is how much people drink. But I've discussed with Henk and Reagan that maybe this bar paradigm may be actually better, and it has been better validated to test alcohol consumption. Namely, people would get a priming drink of a breath that would lead to a breath alcohol concentration of 0.02 to 0.03, and after that they would receive $16, of which they can either purchase 8 alcoholic mini drinks, leading to a maximum of 0.1 breath alcohol concentration level, or just keep the money. So then we can really see, is it the case that those who take the ketone ester, particularly after this priming drink, is it the case that people with alcohol use disorder drink less, or even controls, so they drink less when they are in metabolic ketosis? And third, this bench to bedside proposal that we submitted last July is on a project on brain metabolism and alcohol tolerance using PET methodology. So in this study, we test to the effects, or well, we don't really know if we have received the money yet, it's pending, but to test the effects of metabolic ketosis, again, using this ketone ester on brain acetoacetate, which can be labeled also with carbon 11, as well as brain glucose metabolism in alcohol use disorder. And there we hypothesized that the ketone ester-induced uptake of acetoacetate will be faster in people with alcohol use disorders, just because they're more used to being in ketosis, or at least have higher levels of acetoacetate in their brain. So reflecting enhanced brain uptake of acetoacetate as an alternative to glucose as an energy source in alcohol dependence. And we test the effects of the ketone ester on alcohol tolerance. So what Leandro Vendoscolo showed in his animals that we do not have figures for yet because we still need to study it systematically, but it looked like those who are on ketosis are more sensitive to it, the effect. So I would like to test this in just a fixed dose of alcohol. If you give a fixed dose to people in ketosis or not in ketosis, how do they respond to it? Do they like the effect? Do they crave alcohol more or less, etc., etc., or do they find also the effects more intense? Also the PET scan design developed also with Robert Dude. Thankfully, he was willing to help with this in terms of the metabolites, or just the PET scanning. First, people would get the ketone ester, and then after 30 minutes, we would inject them with acetoacetate. Then we'd have a break, or we scan for 20 minutes, and we'd have a break. And then for another 60 minutes, they would be injected with FDG. So they would be injected with 2 tracers at the same time. And this paradigm has been done in Canada, and it has been shown that a ketogenic diet increases the uptake of acetoacetate and decreases FDG, but it's never been done with a ketone ester. And also very importantly, well, to us at least, is to see how patients with alcohol use disorder respond to this. Is there a faster uptake of acetoacetate compared to those who do not drink alcohol? So yeah, future directions. So very coincidentally, actually, I did not know about this, but Dr. Paco Bravo, who actually runs a study currently on the effects of ketone esters on FDG, but he is particularly interested in the heart. So we've discussed on whether it's possible for us to collaborate on a whole body PET scan and look in the same individuals with or without alcohol use disorder, whether this has an effect on the heart as well as the brain, but maybe we should also work together with liver specialists and see what this ketone ester actually does to the liver. And second, I would be very interested in setting up a larger clinical trial of either the ketogenic diet or the ketone ester on alcohol consumption in outpatients with alcohol use disorder, or actually on alcohol withdrawal in inpatients. It has never been shown that the ester actually has an effect on withdrawal. And lastly, using PMRS, it may be interesting to look at the ratio of NAD over NADH, which is a measure of brain energy. To look at the effects of the ester and also just to test at baseline whether there is a difference in brain energy of this ratio in people with alcohol use disorder versus controls. So thank you so much, particularly to my current collaborators here at Penn, Hank, David, Reagan, and Rose, Jake Dubrov, Bob Mack and Robert Dude, Aaron Schubert. Senhao Shi has started, or he was actually a postdoc in the lab of Daniel Langleben, and he's now working for me, mainly working on functional MRI data. And Tim Pond is helping me right now to do everything, basically. He knows everything. He's fantastic. And still active collaborators, Nora Volkov and Leandro Vendoscolo, George Koob, and Todd King, who was the staff scientist for Richard Veitch, and who has developed this ketone esters, who knows everything about the chemistry, thankfully. Let me know if you have any questions. I'd be happy to answer.
I think we should unmute our audio so that we can clap. That was very nice. And this is actually quite an impressive tour de force on this topic. I have a couple of specific questions, but first I'd like to ask how you came onto this. How did you get onto this observation initially about the ketogenic diet? What was the origin of this for you?
So the origin was really all the FDG findings that we had of
Yeah,
lower brain
yeah,
glucose metabolism, and
right,
then
right.
also
You just
discussing
went from
this
you
with
went from there. I mean, that was
yeah.
very creative. To make that leap is not only been obviously creative, but pretty doggone productive. Have you noticed in the clinical literature that people who go on ketogenic diets for weight loss have a change in their reactivity to or taste for alcohol? Has anyone anecdotally reported that?
Yes, so it has been reported anecdotally, but nobody has studied this. So if you look on PubMed, there's nothing known about the effects of a ketogenic diet on alcohol consumption. But if you read on Reddit, for example, just subjective responses of people who take a ketogenic diet for
Right,
various
right.
reasons, they all say that they cannot tolerate alcohol as much as they used to.
That they describe being almost more sensitive to its overall effects, both sedative and high, or how do they describe it, Corinne?
Um,
Or do
oh,
they?
I would have to look back, but
Yeah, yeah.
there was one person I once spoke to whose wife was on a ketogenic diet once, and they, well, I think after 2 months or so, they started really binging for some reason, for
Yeah,
a
yeah.
baseball
Right, right.
game or something like that. And she said, or she really almost passed out. It was that
Yeah.
terrible, the effect of it.
Huh.
So that's why also we have to be careful, and of course,
Yeah.
there have
Yeah.
to be MDs monitoring this as well, because we
Sure.
don't know yet how people respond to the effect.
Yeah, but it's so intriguing, and the ester does make your life a lot easier than having to
Yes.
do a controlled diet. It sounds like people are better able to tolerate it. But I was just curious about whether it increases the sensitivity to the sedative or the I'll call it the downside of the curve, which is sort of people feeling not the ascending high feelings, but the sort of sluggish down, and whether it's both or whether it's one more than the other in terms of the subjective effects,
Yeah,
which
and I
might
would
have some.
actually really like to try it out myself. Unfortunately, the ketone ester has been sold out for a while because it seems to also have a beneficial effect on COVID-19. So there's suddenly a spurge
Oh,
of people who want to
oh my
use it.
gosh, how
But I
wild.
wanted to test it to see if it also alters my own sensitivity to alcohol.
It's always great to be able to have a data point from yourself in your presentation, like many great scientists in history trying things first on themselves. But I really enjoyed that, and I also think it's really fun to have these parallel studies going on in the animals, where things can be obviously highly controlled and their history, etc. So that seems like that's a very productive path too. I'll shut up and let other people talk, but yeah, that was really fun.
Thanks, Conrad.
Hey, Corinne. Welcome to Penn. It's nice to see you here finally. I had a quick question about your alcohol Q study in your first study. It's actually kind of more of a fun question as opposed to like a real serious one. I'm wondering if you looked at the preference for ketogenic foods versus non-ketogenic foods instead of just like food versus alcohol, and if there's any like group differences. I mean, there's no group differences overall in like the food, so I don't think it would like really affect the results. I'm just more curious.
Yeah, if also halfway through the trial, we were thinking about that. So we have high carb, or no, actually just high calorie food such as hamburgers versus low calorie food versus, or like such as salads. But we actually did not include ketogenic versus non-ketogenic, which was kind of stupid. So that would be definitely something to look at this round.
Yeah.
So no, we haven't tested the effect,
Right.
unfortunately.
Very cool.
Oh, thanks.
Anybody else have some questions for Corinne? I'll ask one while people are thinking. Corinne, just in general, one of the things that we sometimes struggle with is intervals between fMRI reactivity sessions, so that the variable of extinction, some people have more extinction than others when you're in a non-reinforced sort of setting. What are your thoughts about that? How do you address that? What we've often done is tried to design around it so that the first, like if we're looking for the effect of a medication and we want to have the most robust response to our cues, we'll design it so that between groups, the first time people see cues after they're on the medication or the placebo is the first time, and it's more difficult because there can be a repeat extinction effect. So what do you do to get around that, or how do you deal with it? It's something that we've noted when we've attempted to have repeated paradigms. There will be a decline, but it's non-random. Some people have more extinction than others, and then that gets mixed in with a drug effect, right, with repeated tests.
So the first, definitely it's a problem. So the first thing to do is, of course, have a control group. So that's why it was so essential to have this standard American group in there who had the same alcohol background. And also now in this R00 trial to have the order randomized for when to be exposed to the ketone ester and when to be exposed to the placebo. But it's actually fair that after the functional MRI, we would immediately like to expose them to alcohol or to drinking alcohol. So there may actually be an effect of the alcohol cues itself on alcohol consumption, which may be stronger in those who drink alcohol regularly compared to those who don't. So that's probably a limitation. I would ideally not do it on different days though, because they already come in 3 times now. I'd rather not let them come in 5 times, but
Yeah, it's a challenge. I mean, I think I often, when we go into review with a paper or a grant, people talk about the joys of doing repeated Q activity and the challenges of it. And the challenges for me have often sort of loomed. And so I've tried to design around it, but I'm always wishing that there was a way. But counterbalancing works as long as there's still something to counterbalance for. But if there's a huge extinction effect, then you have a challenge in that the second or third exposure will be less even for the control group, and at some point it's going to be potentially squeezing the groups together, And so,
or something else maybe to do, what you always do, is to look at unconscious cure activity.
Yeah. I try to do that too, and it might be that that's more robust or would be less affected, but I think that there's, it's a possibility of having extinction outside awareness. Always we can talk about that, but anyway, I was intrigued by that too, because I saw those repeated administrations and I felt excitement to be able to think that they could be useful and that you could do it and still have a robust response. And I guess the other way would be to have something that actually would be an occasional, like with a drug like alcohol, you could have some occasional alcohol so that the cues essentially are signals and they maintain their reactivity because there is occasionally an alcohol trial, following the cue. I mean, that's another paradigm, but just dealing with the way. I'll shut up again. I will. I promise. Someone else, please.
I had a question. I thought it was a great talk and it was really exciting. I don't know if I missed what the full battery was of neuroimaging that was done and whether it was done pre and post. I know you've mentioned several pieces, including a functional scan. You looked at some structural findings. I was wondering if there was also resting state by any chance?
So we have actually collected resting state and we haven't looked at it yet. So that is definitely something to look at it next. And also we had a visual attention task, which I haven't looked at yet. And also just for the scope of presentation, there was also a whole battery of cognitive tasks that people did in week 1 and week 3. There were not really strong effects of the ketogenic diet. And we also measured sleep, and there seemed to actually be a counteractive effect, namely those on the ketogenic diet seem to have actually less sleep. So that's going to be the next thing to write up as an extra finding.
Interesting.
Can you give me your last name? What is your book saying?
Uh-oh. This may be some PII being
Do we have any other questions for Corinde today? How are you going to do all this work, Corinde? This is a lot of wonderful stuff. It's a lot. Great. And with COVID.
It took me 2 months to actually move to Philadelphia, but now I'm here, and especially with Tim helping me to set up the IRB, I think we will somehow do it.
Of course you will, but it's aspirational, and I think it's wonderful. Other comments or questions?
I'd like to ask Corinde. First of all, it was a beautiful presentation, and it's got me thinking because we're in the middle of a epidemic of type 2 diabetes, and we do check for ketogenic means as a part of clinical care, but normally we don't do serial measures the way you did. And so, is there other data that correlate in diabetics who have elevated levels anyway? Any effect, because the coexistence of alcohol use disorder and diabetes is very common, but I don't know anyone studying it.
I don't know either. That's a fantastic suggestion. Do you maybe have actually patient records that we can actually look at levels of ketosis, or if people are on ketosis or not, who have either diabetes or alcohol use disorder or both or none? It may be interesting to look at it retrospectively.
Yeah, it's done for reasons of determining toxicity in the clinic. But typically, you want it to be low, and you don't. If it's not elevated, you don't follow it. So, but maybe someone has. I mean, it just seems to me that another way of looking at it is that it could be added to the regimen of a person with alcohol use disorder that you're trying to get on a medication like naltrexone. Would like to get them detoxified in a stable way before you start the medication, and it could be, in a practical way, it could be very useful.
Yeah, that's a fantastic suggestion. But I honestly do not know whether anyone has studied this.
So don't drink a lot of fluids for 2 days before I see you, so as to avoid giving a dilute urine. Okay, all right. Thanks for calling. I'll see you later.
So busy.
We're just still busy taking calls. Do we have any other questions for Corinde? I see Aaron Schubert has a chat for you or for the group that Paco Bravo is collecting ketone levels in his current FDG study, and he's identified a point of care. So he's hoping to study diabetic patients as well as healthy controls. So there you go. Thank you, Aaron.
Yeah, Aaron has actually put me in contact with Paco, and he sends us his, or she actually sent me his protocol. So it's fantastic if we can work more intensely together on this.
Sounds like you've had no trouble finding a bunch of collaborators here, and I think that's wonderful. Even
well, I've already noticed how collaborative the people at Penn are. So
even during a pandemic. Well, if we have no more comments or questions, we will allow people to go back to their quiet activities. And Corinde, thank you so much. The talk has been taped, so for people who couldn't sign on today that we'll let them know. It'll be posted in a few days, and it'll be on the website. And Corinde, you can refer all your friends and family to it. Okay? Talk to you all later. Thanks again. Bye-bye, everyone.
Thank you.
Great job.